Okay, three types of muscle: skeletal muscle, smooth muscle, and cardiac muscle. Okay, this is smooth muscle. Here's some smooth muscle out here. This is a thick-walled, medium-sized artery. What kind of tissue is right here? And here. Dense, irregular connective tissue. Then we have endothelial cells in here. You can't see them very well. See all these nuclei in here? Those are the nuclei of the smooth muscle cells. Okay? This stuff in here is blood. Here is a vein in comparison to an artery. Here's a smooth muscle in here. It's really thin in comparison. Okay? And of course you have endothelial cells in there, that inside that you can't see too well, except maybe over here. See this stuff right here, this clear stuff, that area that's clear? That's called artifact. Okay, artifact is a result of the tissue separating during the fixative process. What do you do when you do that? First, you subject the tissue to increased concentration out. Start at 10%, 20%, 30%, 50%, 75, 80, 90, and then 100% alcohol. Why do you do that? To remove the water. So you're really drying the tissue out in alcohol. And that causes it to distort. And then you embed it with paraffin. It's like beeswax, okay? Some of it's even has more consistency than beeswax. And then you embed it in that, okay? And that's why you take the water out, because then you can embed it. You embed it, and then you take really thin slices. And all those steps, you're actually putting tears in the tissue, and we call it artifact. Okay? So this over here is skeletal muscle. Okay? So you've got arteries and veins going through skeletal muscle. Check. What do you think these are? Those are cross-sections of nerves. Arteries, veins, and nerves travel in the same area. It's like how you wire a house. Well, your wiring follows the plumbing. Okay? And the plumbing, and sometimes the wiring follows the conduits of your heating system. Okay? Makes it easier that way. And that's what's happening in you. Okay, higher power of the same thing. Here's your smooth muscle. Here's kind of like a connective tissue capsule. And here's the artifact. So this right here is supposed to be stuck to this. It just separated. Here's a smooth muscle of the vein. Here's the red blood cells. Okay? This is a funny slide because the technician that made this slide cut the tissue really thick. And it gives it this appearance. This is more smooth muscle. Okay? But this is smooth muscle in your GI tract. Because here's some mucosa up here. Here's some dense irregular connective tissue that's part of the submucosa. Here's a circular layer of smooth muscle, and here's a longitudinal layer of smooth muscle. So you have two layers of smooth muscle around the tube called your intestines. One is circular, and one is running this way along the tube. So that's why you have smooth muscle that looks like this, because it's cut longitudinally, and this smooth muscle over here is cut cross-sectionally. But it's all smooth muscle, okay? So it's like the difference between cutting a cell that's shaped like this with a nuclei in it. Like this would be the cut, or like this would be the cut. Okay? That's basically what's happening. Okay, here's some more smooth muscle. This is a different technician using different state, and this technician cuts the tissue even thinner. Okay? So this is circular layer. Here's some more circular layer. Here's the mucosa down here. Here's the longitudinal layer out here. And there's a lot of artifact. Look at all this artifact. All over the place, all these cracks. This is a collection of nerve cell bodies between the two smooth muscle layers. We'll deal with that later.
What is the definition of ganglion?
That's a ganglion. The definition of a ganglion is a collection of nerve cell bodies outside the central nervous system. That right there is a ganglion. This is a ganglion. So they're sending axons out to the smooth muscle so it can coordinate peristalsis. Okay?
Artifact relating to muscle?
It's everywhere. Every tissue is going to have some.
So what makes the ganglion cyst then?
Makes the ganglion what?
Ganglion cyst. That's five things.
That's when you cut a nerve and you make this cyst. You want to feel one right here?
Yeah.
Okay, there's a little, yeah, it's right there.
So you,
Is what happens is I cut a nerve, and the nerves, if you don't have the connective tissue surrounding the nerve, it like forms this little bump. That's what that is. They're benign. Okay? And they're really not ganglia. They're just a bulge of axons trying to find where they're supposed to go because they don't have surrounding connective tissue. They don't know where to go. So they like do little loops all over the place that make this. Okay, here is a cross-section of somebody's intestines, probably a mouse. So here's all your mucosa in here. Here is the simple columnar epithelia. The lumen would be in here. That's not our fat. Then you have circular smooth muscle and longitudinal smooth muscle. Here's a higher power of the same thing. Got it? And out here will be a single cells. That's the viscera serosa. Okay? You finding your way around? Okay, higher power, the same thing. What can I say? Smooth muscle, circular layer, longitudinal layer, visceral serosa, simple columnar epithelia of the mucosa. Non-keratinized stratified squamous epithelia in somebody's esophagus. Okay? Here you have smooth muscle here and smooth muscle here and another layer of smooth muscle. And the reason why you have three layers in the esophagus, at least in this area, and I'm not showing it in this, is because you have two muscle types in the upper esophagus. The upper 1/3 of the esophagus is skeletal muscle and smooth muscle. The lower 2/3 is nothing but smooth muscle because you swallow. It's intentional. And then you start the peristaltic wave, and that's unintentional. Okay? So this is smooth muscle going in all kinds of different directions. That's what happens in the uterus. The uterus is a mass of smooth muscle. Okay? And you see, here's an artery right here, and you have the artery smooth muscle going in a circle, and the little endothelial cells. See them? This is low magnification, high magnification of the same artery. And here's more smooth muscle, more uterus probably, at higher magnification. Here is cardiac muscle. Okay, here it is under low power, here it is under high power. You see the striations, the intercalated discs. Okay. Now, in some cardiac muscle preparations and some skeletal muscle preparations, the technician didn't use a sharp enough knife, or the technician was just lazy and didn't do a good job. And as a result, sometimes you will not see the striations, and sometimes you will not see the intercalated discs. The intercalated discs of cardiac muscle are holding the cardiac muscle cells together, and there's gap junctions in there. And a gap junction is an electrical synapse of large electrical impulses to travel from cell to cell. Okay?
Is that what the vertical lines are? The one on the, well.
These lines right here are the striations.
Right.
These thick lines are the intercalated discs. Okay? Here is more cardiac muscle. And you can tell it's cardiac muscle because you can see some intercalated discs. You can also tell it's cardiac muscle because the nuclei are oval, not flat. Okay? And the rest of this cleared area out here, that's artifact. Here it is under low power, here it is under high power. So if you look at this under low power, you really can't tell what you're looking at. You see it under high power. Now keep this in mind. The striations and intercalated discs are really, really, really small. So you have to have your microscope tweaked really well because you can go just up out of the plane of focus in either way, and the intercalated disc and the striations disappear. See them. So you got to tweak it. Okay? Everything has to be right. The condenser has to be up and back down a little bit. The condenser has to be closed and opened a little bit. Okay? And then you have to tweak the rest. If you don't have those two maneuvers done, you will not see striation, even if on a good slide. Sometimes the slides aren't so good and you can't see them anyway. Here is more, another preparation of cardiac muscle. Here's the striation, or the intercalated discs, and the striations, and the oval nuclei. This is cardiac muscle cut and cross section. The other images, you saw them on longitudinal section. Cross section, you get the nuclei like in the middle of the cell. And all this separation basically is just artifact. Here's some smooth muscle of a coronary artery with the endothelial cells in it. Okay? This is the smooth muscle out here. We'll talk about the pieces and parts of an artery later. This out here is artifact. What kind of tissue is this? You see striations. So your choices are two, cardiac muscle or skeletal. The nuclei are flat, skeletal muscle. There's no intercalated discs, skeletal muscle. Okay? Low power, the striations start to disappear. Higher power, they come into focus. This is skeletal muscle cut on cross section. Remember I showed you in this image, the nuclei are in the middle and they're oval. The nuclei are flat and on the periphery. Okay? On cross section. Pretty slick, huh? What can I say? That's about it. Okay, more skeletal muscle. Now this right here, you may see something like this that kind of like looks like an intercalated disc, but it's not. It might be a smashed nuclei or lint or dust on the slide. Okay? But you can see the striations. That's it. Sure.
The serosa of the intestine, that is serosa, right?
The intestine, the serosa is simple squamous. Only on the ovaries will you have simple columnar as the visceral serosa. Okay? Okay.